Thank you, Dr. Kemmerett, for the introduction. My name is Cassie Newman, and I am a second-year PhD student at NC State. And today I will be presenting my work on a de novo Virginia-type peanut genome assembly and whole genome sequencing panel. I would like to start with a brief introduction to NC State's peanut breeding program. We work exclusively with Virginia-type peanuts, and those are characterized by having large kernels, and they are used for the in-shell market. Here I have a very generalized schematic of our breeding program. So on the left, we have a standard program that we use that utilizes single plant selections. And on the left, we have an advanced program that uses a winter nursery to achieve 2 generations per calendar year. And they come together in our advanced trials. And I'm showing you this today because there's something missing here historically, and that is the integrated use of genomic tools. So that's what I'll be addressing in my project. So what is available to us now in terms of these tools? The diploid progenitor genomes were released in 2016, which opened the gate for several tetraploid peanut genomes to be released in 2019. And I would like to point out that Virginia-type peanuts are not represented in this collection. As for genotyping technologies, I would venture to say the 2 Arachis arrays are the most prevalent in the peanut community as of today. And a previous PhD student at NC State took 200 Virginia-type lines and had them genotyped on the 48K SNP array. When he got his data back and filtered it down, he was left with less than 2,000 of the 48,000 SNPs that he could actually use for that collection of genotypes. So that means that the arrays are not the most efficient strategy for our program to use in terms of genotyping. And that's what drove us to develop our own tools from scratch. So the central question is, how do we go about integrating genomic tools into applied Virginia-type peanut breeding pipeline? And that's a very complex question that needs to be informed by good background information. And what my project does is provide that background. So I will be answering the 2 questions in orange. So can we develop a Virginia-type reference genome? And how many markers can we obtain that are informative to our program? So the hypothesis is, if we utilize the latest sequencing technology to create a Virginia-type peanut genome and genotype important germplasm, we will have enough information that we need to be able to create these genomic tools for our program specifically. So starting with my materials for the Genome Assembly, we used Bailey 2 to represent Virginia-type peanuts. And Bailey 2 is a near isogenic line to Bailey, and Bailey is a very widely accepted and planted cultivar in this growing region historically. The difference between Bailey 2 and Bailey is that Bailey 2 has the high oleic trait, and Bailey 2 is shown here. To start the project, I received long read, short read, and optical mapping data. Here are my methods for the genome assembly. So if you look at the bottom of the slide, you can see how I took the raw sequencing data and transformed it through a series of steps in order to create a chromosome scale assembly. And each of those points corresponds by color to the chart above. And the chart is showing how many the number of scaffolds on the y-axis. So how many pieces or scaffolds is the genome made up of at that point in the assembly process? And underneath, we have the max scaffold length. So of all those pieces, if you're looking at the biggest one, how long is it? So you can see as I go through the process of joining those pieces together, my total number of pieces gets smaller and the size of the largest piece is growing. Moving on to the materials and methods for my whole genome sequencing panel, we collected 94 lines that represent germplasm of historical or agronomic value to our program. And the composition of that panel is shown in the pie chart here. So I received paired end Illumina data at an average of 8x coverage for all of these lines. Moving on to my results for the Genome Assembly. So on the left, we have a basic assembly metric table. And again, what those first two columns are showing is how many contigs or pieces the genome is made up of. And if you sum those pieces together, what is the total length of the genome? So what this is showing is that I was able to achieve the same basic sequence total, but I did that with fewer number of contigs. So that shows that Bailey 2 is a more continuous genome than the accepted standards. As for the last column, that's showing the BUSCO score as a percentage. And the BUSCO score represents how complete the genic portion of the genome is. And you can see that Bailey 2 is performing as well as the previous standards there. Moving to the right, we have a whole genome alignment between Bailey 2 on the x-axis and TipRunner version 2 on the y. The red color is showing when the genomes are aligning in the same direction, and the blue color is when the two genomes are in the opposite orientation. So what this is really showing is that the TipRunner and Bailey 2 assemblies are largely collinear. So it's novel that we have a Virginia type reference, but what does that actually do for people using it? So I did a short read alignment study that used both Bailey 2 and TipRunner 2 as reference genomes. And I took short read data and aligned it to both of those references. And I looked at how well the short read data aligned. And the results are shown here in the bar chart. So alignment rate as a percentage is on the y-axis, and the lines that I had short read data for are on the x-axis. TipRunner is in tan, and Bailey 2 is in brown. And you can see that Bailey 2 is performing well in relation to TipRunner across the board. And I will note that the alignment rate change between the two genomes is very small, but when you're talking on an order of millions of short reads, that small change in alignment rate can actually make a difference in how well you're able to use your data. Moving on to the whole genome sequencing panel results, I did a variant discovery pipeline and I received back 16 million sites. And then I filtered that down very strictly, and I was left with 14,000 of the highest quality sites. So again, that represents the bottom end of how many sites we can have across the genome. On the right, I have plotted chromosomes 1 through 3 and how those SNPs are distributed throughout the length of the chromosome. So the SNP count is on the y-axis, and the genomic coordinate is on the x-axis. And you can see that we have a pretty even distribution of variants across the genome. And that's great because a goal of our program is to create genome-wide markers. So with this, we can see that that's actually possible. So it is a tremendous accomplishment of our program to have a Virginia type reference genome and have genotypic information on almost 100 lines of interest. But what can that collection of data actually do for an applied breeder who's trying to release superior cultivars? So I thought I would include an example of the use of this data. So this example revolves around the now famous wild species, Arachis cardinacei introgression into the tetraploid hypogaea genome. And that event is shown at the top of the pedigree here. And I'd like to note that segments coming from cardinacei actually confer important resistance to peanut, such as resistance to the fungal pathogens that cause early and late leaf spot. So this event has an economic and agronomic value to the peanut as a crop. So you can see how the modern Virginia type cultivars in that salmon color all have the cardinacei introgression in their background. And the different boxes I've colored, we're going to be talking about on the next slide. So again, the salmon colored box represents modern Virginia types that should have that introgression event. The mustard colored lines are historic Virginia type cultivars that should not have the introgression. And the tan lines are lines that were included in our whole genome sequencing panel, but they have not been used as parents in our program historically. So as promised, I've carried that color scheme through to this slide, and we have the lines that we just talked about on the y-axis, and we're looking across chromosome 2, chromosome 8, and chromosome 13. And we're just looking at the segments that are known to have those Cardinacii introgression events. And the coordinates are shown on the bottom of each of these chromosomes, and they're relative to Bailey 2 as a reference. The red color represents the homozygous Cardinacii allele. The dark gray color represents the homozygous alternative allele, and the gray color represents a heterozygous call. So on chromosome 2, you can see that the historic Virginia types don't have the block. The modern Virginia types do have the block, and the IAC and SPT lines do. And I'd like to note that you can actually see recombination happening inside of that block for both Emery and IAC 322. Looking at chromosome 8, we can see the same phenomena happening where the historic Virginia types do not have the block. The modern Virginia types do have the Cardinacii block. IAC does not have it, and SPT does have it. Moving on to chromosome 13, we actually see something novel happening where none of our Virginia type cultivars have the Cardinacii block. However, the IAC and SPT 1012 lines do. So this represents an extraordinary opportunity for a breeder to place markers within those Cardinacii segments and backcross them into our lines in order to stack all of those segments into one line. So this is an example of just how much information we've gained from this study. And again, we're just looking at a handful of lines out of the total that have been sequenced. And I'd also like to note that SPT 1012 OL has all three of those segments already within itself. So that's really important to note. So circling back to our central questions, can we develop a reference genome specific to Virginia type peanuts? Yes, we have developed Bailey 2, and it is a reference level genome. And how many markers can we obtain that are truly informative to our program? And here we found a minimum of 14,000 markers that we can use. So moving on to our future directions, we are actually designing Cas markers that span and are interspersed within those introgression blocks for marker assisted selection. And we are doing marker development for genome-wide characterization of our breeding lines. And we're going to be testing various genotyping and bioinformatics strategies for that project. And with that, I'd like to say thank you to everyone who made this research possible, and I'm open to taking questions.
Thank you very much for that presentation. And we have here a comment from Dr. Soraya Bertioli. Beautiful talk, she says. We have observed introgressions from A. cardenas. I'll tell you what, I'm gonna have to somebody help me pronounce that. You know what I'm talking about, the card one on IAC 233 and SPT should be in chromosomes 03, not 13. How do you explain this difference?
Okay, so I was, okay.
And there's two parts. You answer that part, now come to the second part.
Sure. So I didn't explain the background for this introgression event from Cardinacii in my slides, but it was taken from crossing a diploid by the tetraploid, getting it to a tetraploid, a sterile tetraploid state, and then being doubled up to a hexaploid state in which it was selfed for many generations and then reverted back to a tetraploid at some point. So there was a lot of opportunity for the chromosomes to be matching in different ways than they normally would during that introgression process. And that is one reason why we might see this on 13.
Okay. Second part of that question is, based on the published work of the genotyping of the US peanut mini-core collection, how much difference do you expect to see between Bailey 2 sequence and TIF runner 2 sequence?
Here, can you repeat that?
Yeah. Based on the published work of the genotyping of the U.S. peanut mini-core collection, how much difference do you expect to see between Bailey 2 sequence and TIFF-Runner 2 sequence?
Sure. So I'm not sure exactly from that study how much difference was found from the mini-core collection. However, I did create those whole chromosome alignment plots. And we can actually see that there's not much structural variation between the two genomes here. So I guess as an answer to that question, I would say that there is relatively little difference that we did observe in practice between the two genomes, besides some of those introgression regions.
All right, that's the time we have. Thank you very much, Cassie, for joining us today. Very nice presentation. Our
Thank
next
you.
speaker will be Alan Pepper.